Hello, good day to you. I am S.O PienS. I want to give a presentation which titled Cell Physiology. Now it's going to involve a series of presentation on physiology. As I discuss cell physiology. I think we'll discuss other fields of physiology. Okay, so now before I proceed, let me give you the highlights of this presentation. Number one is definition of a cell. Number two, organization of life. Number three, structure of a cell. Number four, cell junction. Number five, cell transport. Number six, cell division. And number seven, the importance of cell physiology in other fields of physiology. Now, to start with the first one, which is a definition of a cell. Now, the cell is the basic structural and functional unit of life or existence. In other words, that definition has two major keywords. That is the structural unit and the functional unit. Now, the structural unit because it has the typicality of every structure found in a living organism, and is a functional unit because it can perform all the functions that a living entity will perform, and so a cell is an independent unit of life or existence. Okay, now talking about the organization of life, you need to understand that atoms come together and they give rise to a molecule. Molecules come together, they give rise to a macromolecule, such as proteins, lipids, carbohydrates, and nucleic acids. Your macromolecules come together, they give rise to organelles. Organelles come together, they give rise to a cell. Cells come together, they give rise to a tissue. Tissues come together, they give rise to an organ. Now, organs come together, they give rise to organ system. Organ systems come together, they give rise to multicellular organism, e.g. human being. Wow. So that means you can still trace back your existence to anatomical level. Alright, now going to the third point, that is talking about the structure of a cell. Of course, we are talking about eukaryotic cell. Now talking about the structure of a cell, we can talk about two things. That is looking at the cell membrane and the contents of the cell. Now talking about the cell membrane being an organelle which has some macromolecules such as the proteins, the lipids, and carbohydrates. Now looking at the protein portion of cell membrane, it has two major types. You have the integral and the peripheral proteins. Now the proteins, they have a number of functions such as serve as channels, serve as pumps, receptors, carriers, enzymes, antigens, and in cell adhesion processes. Okay, then they are also involved in the maintenance of structural integrity of this cell membrane. Now looking at the lipid portion, the lipid consists of the phospholipid bilayer and the cholesterol. Now the functions are majorly for selective permeability of substances across the cell membrane. That is, it allows only lipid soluble substances to actually penetrate through the barrier, while the cholesterol is involved in the regulating the fluidity of the cell membrane. Okay, now looking at the carbohydrate portion. For the carbohydrate components of the cell membrane, it's in combined state as glycolipids and glycoproteins. Now the functions are, because they are negatively charged, they repel negatively charged substances, and so they are involved also in selective permeability. Then they are also involved in cellular communication, that is in cell adhesion. Then they are also involved in immune reactions, that is the role of glycocalyx in cellular identity. Glycocalyx is a complex of glycolipids and glycoproteins. Okay, then also the carbohydrate portion is also involved in the serve as receptors. Serve as receptors. Then now looking at the contents of the cell. Now I've talked about the cell membrane, so we've considered the composition and the functions of individual macromolecule present in the cell membrane. Before we look at the contents, let's consider these terminologies. Now we have protoplasm. Protoplasm is all the content of the cell by the time you penetrate the cell membrane. That's all the content, that is the cytoplasm plus the nucleus. That's what forms your protoplasm. Cytoplasm is a compartment of the cell excluding the nucleus, while vacuoplasm is the portion occupied by the cellular organelles inside the cytoplasm. The cytosol is actually the fluidy portion of the cytoplasm. Nucleoplasm is actually like the cytoplasm in the nucleus, while the hyaloplasm is the counterpart of cytosol in the nucleus. Okay, so the portion that is not occupied by the nuclear suborganelles, that is what forms your hyaloplasm. Hyaloplasm is HYALO plasm. Okay, now let's consider the cellular organelles. Now the cellular organelles, starting with ribosomes. Ribosomes are involved in synthesis of proteins. The rough endoplasmic reticulum is involved in synthesis of proteins and also degradation of worn-out organelles. The smooth endoplasmic reticulum, which is also called the sarcoplasmic reticulum in muscle fibers, it's involved in lipid synthesis, storage of calcium, like in skeletal muscle fiber, detoxification functions such as microsomes in the liver hepatocytes. Okay, then we also have Golgi apparatus. Golgi apparatus has two surfaces, the cis and trans surfaces. The cis is facing the endoplasmic reticulum, while the trans surface is facing the cell membrane. Now the Golgi apparatus is involved in processing, packaging, labeling, and final makeup for the synthesized proteins and lipids, that is from the rough and smooth endoplasmic reticulum. The lysosomes is involved in intracellular digestion of worn-out organelles and also macromolecules. It's also involved in secretory functions and is processed from Golgi apparatus. Lysosome is in two forms. We can have primary and secondary lysosomes. The primary is the free lysosome, while the secondary is actually the active or attached lysosome. That is the one that bind with endosomes. Okay, then the next organelle is peroxisomes. Peroxisomes are actually pinched off from endoplasmic reticulum. Peroxisomes are involved in breakdown of peroxides in fatty acids and purine metabolism. They make use of oxygen. They are involved in formation of myelin as in neurons, and also in bile acid formation as in liver cells. Then the next cellular organelle is centrosome. Centrosome consists of two centrioles, which is involved in cell division and also the movement of chromosomes. Then we also have mitochondria, which is the powerhouse of the cell. It's involved in ATP synthesis. It has a role to play in apoptosis. Then we have cytoskeletons. Cytoskeletons such as microtubules, microfilaments, and intermediate filaments. These cytoskeletons, they are involved in maintaining the shape of the cell and its movements. Then finally we look at the nucleus. Nucleus is a major organelle in the cell that controls the activity of the cell. It's involved in formation and storage of nucleic acids such as DNA and RNA. Then it has a role to play in cell division and also protein synthesis. Before we actually go on looking at the cellular organelles, let's talk about some of these nuclear structures. Chromosomes are deeply stained strands found in the nucleus. Then DNA is inside chromosomes. DNA forms complex with histone proteins to form nucleosomes. Then DNA with histone proteins with non-histone proteins, they actually are together inside chromosomes. Genes are also the basic unit of heredity, also located inside DNA, a segment or a portion in the DNA, and they are the basic unit of heredity. Now, somatic cells contain 46 chromosomes, while gamete cells contain 23 chromosomes. And so we say somatic cells, they have diploid number of chromosomes, while gamete cells or reproductive cells, they have haploid number of chromosomes. Okay, so we go to the next topic, which is the cell junctions. The cell junctions and communications. Now, looking at this cell junction, this is talking about the interaction or the communication between cells in the body. Okay, and is usually found between epithelial cells and some other cells, but majorly epithelial cells. Now, we have two major classes of cell junctions. We have the anchoring junctions, we have the communicating junctions, and we have the occluding junctions. The anchoring junctions consist of the desmosomes, the adherent junctions, the hemidesmosomes, and the focal adhesions. Now, the desmosomes and adherent junctions are between cells, usually epithelial cells, and hemidesmosomes and focal adhesion, they are between cells and basement membrane. Okay, now for the communicating junction, it involves the gap junction and chemical synapse. Gap junction as in electrical synapse, that's in cardiac muscle fibers, and also in smooth muscle fibers in the GIT. Then we also have chemical synapse, which is also a form of communicating junction that exists between neurons and neurons, and neurons and muscle fiber, that is at the level of your neuromuscular junction. Then finally, looking at the occluding junctions. Occluding junctions are actually they are majorly the tight junction, which we also call zonular occludens. And this tight junction is actually involved in selective passage of molecules. It's highly selective in allowing passage of molecules. So it is present in the intestine between enterocytes, and also in barrier systems like blood-brain barrier, blood CSF barrier, blood nerve barrier, and blood testis barrier. Okay, then let's briefly look at these cell adhesion molecules. Now, these are molecules that are actually involved in cell adhesion processes. Now, we have integrins, we have cadherins, we have IgG superfamily, and we have selectins. So you can acronymize it as ICs, that is ICIS. Okay, then that is all for these for that aspect. Now, looking at the fifth topic, which is cell transports. Now, cell transports is a process that involves the movement of substance across the cell membrane. So, having understood the physiology of a cell membrane, now we can easily understand the mechanism of transport across the cell. Now, basically, we have two major mechanisms that are involved. Now, we have passive and active transports. Okay, for passive transport, we have simple diffusion, we have facilitated diffusion, we have osmosis, we have filtration, and bulk flow. Okay, these are actually under passive. Now, passive because substances moving across the cell membrane is moving down the concentration gradients, and it does not require energy. Okay, now simple diffusion is actually diffusion across the lipid bilayer. Facilitated diffusion can be in two forms. We can have carrier mediated facilitated diffusion, we can also have channel mediated facilitated diffusion. Now, your carrier mediated can be, for instance, all these transports of all these proteins like sodium independent glucose transporter. That's an example of carrier mediated. Okay, while example of channel mediated facilitated diffusion can be all this transport of water molecules via aquaporins. Okay, now the channels can be gated or ungated, that is, can be regulated or unregulated. So gated channels can be voltage, you can have voltage gated channels, you can have ligand gated channels, and you can have mechanically gated channels. Okay, before we proceed, we need to understand some basic terminologies. When we talk about concentration gradient, electrical gradient, and electrochemical gradient, Concentration gradient is the gradient created due to changes or differences in the concentration of substances in two different compartments. Electrical gradient is a gradient based on the differences in the charges of substances, while electrochemical gradient is a combination of electrical gradient and concentration gradient. So you need to note those terminologies. Then looking at osmosis, which is still under passive transport. Osmosis is the movement of water molecule from the region of higher water concentration to the region of lower water concentration through a semi-permeable membrane. That semi-permeable membrane is actually selective in actually allowing the passage of water molecule alone to actually pass through. Then filtration is the movement of substance that involves hydrostatic pressure. Movement of substance from the region of higher hydrostatic pressure to the region of lower hydrostatic pressure. A typical example is the glomerular filtration you have at the level of the renal corpuscle, that is in the kidney. Then finally, under passive, we talk about the bulk flow. Bulk flow is the massive movement of substance across the membrane or barrier, and a typical example is in the respiratory membrane. Transport of respiratory gases across the respiratory membrane is a typical example of bulk flow. Then before we leave passive transport, let's quickly look at some factors affecting rate of diffusion. We have a number of factors that affect rate of diffusion, such as concentration or electrical gradient across the cell membrane, membrane permeability, temperature, charge of the diffusing substances, size and shape of the diffusing molecule or substance, the solubility, thickness of the cell membrane, and distance of diffusing substance to the membrane. Okay, so those are the factors that actually affect the rate of diffusion. Let's look at the second mechanism of transports. We have active transports. Now, active transports can be divided into 4 forms. Now, active transports can be in 2 major forms. We have primary and secondary active transports. Then we also have endocytosis, exocytosis, and a special form of transport that we call transcytosis. Now, for primary active transports, this actually requires direct usage of energy in form of ATP. You have some examples like sodium, potassium, ATPs, like hydrogen pump, calcium pump, and so on and so forth. Then for secondary transport, we have indirect usage of energy that is usually got from the primary active transports. Take for instance the co-transports of sodium and glucose across the cell membrane. Now, your secondary active transport can be in 2 forms. You have co-transport and counter-transport. Co-transport is 2 substances moving together in the same direction. Counter-transport is moving in opposite direction. Then you have protein carriers. You have proteins that are involved in the transport that we classified as unipods, sympod, and antipod. Unipod is simply a protein that can carry just one substance. A sympod can carry 2 substances in the same direction, while antipod carries 2 substances in different directions. Endocytosis is a special form of active transport that is in 3 folds. We have phagocytosis, pinocytosis, and receptor mediated. Phagocytosis is actually the engulfment or the invagination of massive substances such as bacteria, and that can easily be depicted in most of these phagocytes in the body, that's the immune cells. Pinocytosis is involved in cell drinking, such as uptake of proteins. Then receptor mediated is in different forms, involving different receptors like clathrin, receptor mediated, like caveolar receptor mediated, and so on and so forth. All right. Then another form of active transports is exocytosis, which is actually opposite of endocytosis. Now, exocytosis involves the uptake of exocytosis involves the release of cellular contents, such as release of some proteins out of the cell. Okay. Then another special form of transports that involves active mechanism is actually what we call transcytosis or vesicular transport or cytoempsis. Now, transcytosis actually involves both endocytosis and exocytosis, in which case now it involves the transport of substance across the cell membrane. When we say trans means across, so like the absorption of nutrients in the GIT, in the enterocyte, that is movement from the luminal membrane to the basolateral membrane. Okay, at the level of luminal membrane, it involves endocytosis, that is the uptake of the substance, while at the level of basolateral membrane, it involves exocytosis, that's release of the contents from the enterocyte into the interstitial spaces, then into the blood. Okay, that actually ends the cell transports and its mechanisms. Then we look at cell division. That is the sixth topic. Cell division. Now, majorly we have two forms of cell division. We have mitosis and meiosis. Your mitosis take place in somatic cells, while your meiosis take place in gamete cells. And mitosis is actually multiplication division, while meiosis is a reduction division. Your mitosis consists of prophase, metaphase, anaphase, and telophase, PMATs. While your meiosis has two forms. You have meiosis 1 and meiosis 2. Okay, there's a difference between cell division and cell cycle. Now, cell cycle is a series of events that take place throughout the lifetime of a cell, and it involves interphase and mitosis. Interphase consists of G1 phase, S phase, and G2 phase. Now, between S phase and G1 phase, you have a resting phase that we call G0 phase. G1 phase consists of growth and development. S phase consists of DNA synthesis, while G2 phase is associated with preparatory phase for cell division. Okay, so G0 phase is actually the resting phase where some cancerous cells do not enter the G0 phase. Okay, then finally, before I go on this presentation, let's look at the importance of cell physiology in other fields of physiology. Now, finally, before I end this presentation, let's look at the importance of cell physiology in other fields of physiology. Now, you understanding cell physiology will actually help you to understand other aspects of physiology. Okay, other aspects of physiology such as nerve and muscle physiology. When you talk about nerve and muscle physiology, you are looking at cells, neurons, muscle fibers. So you look at cells and subcellular structures. When you look at blood physiology too, you may talk about the blood cells. When you look at GIT, it's a function of enterocytes and some other glandular cells that are involved in secretions. So when you look at endocrine, look at renals, look at reproductive system, look at respiratory physiology. Now, you discover that all these systems in physiology are actually associated with cell physiology, and you understanding cell physiology will actually give you the basic understanding for you to understand other fields of physiology. Then finally, before I go, you need to understand that cells in the intravascular and extravascular systems, they are suspended in an environment called internal cell that we call extracellular fluid. Extracellular fluid is divided into interstitial fluid and the circulating blood plasma. Fluid inside the cells is called intracellular fluid, ICF. Then fluid in small quantity located in confined spaces and cavities of the body, they are collectively known as transcellular fluid. Now, we have about 75 to 100 trillion cells present in the human body, and each of these cells has a role to play in homeostasis. So I hope you've actually enjoyed this presentation. Watch out for other series of this presentation. Thanks for listening, and God bless you. You will succeed.